All right, so we're going to start by talking about the chloroplast, which, if you remember, is the site of photosynthesis within the cell, and it has two membranes. Why does it have two membranes?
Because of the symbiosis theory.
Okay, good. Endosymbiosis theory is that it was once its own organism, and then it got captured by a larger eukaryote, right? So you've got two membranes. It actually has another membrane, and that's what we were referencing in the quiz. And this membrane wraps around itself like this. Okay? And so that membrane is called the thylakoid membrane. All right, and one little turn of that makes this little flattened disc-shaped thing, and this is called a thylakoid. And then they stack together to form a granum. Okay, this is another just poorly drawn one. Okay? So if you have multiple granum stacks, they are called grana. Okay, so plural is grana, singular is granum.
Wait, so the box and noodle are...
They have the same thing. Yeah, I just didn't want to... I'm lazy. Okay, and then they have little connectors between them. And these are called the stroma lamellae. 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 Okay. And then within a thylakoid, then, you have, I'm going to zoom this in, the thylakoid space. And then outside you have a fluid, and it's fluid inside as well. Outside the fluid is called the stroma. So kind of like the cytoplasm is what the organelles are floating around in the cell. The stroma is what's surrounding all of the stacks of granum, or the grana, within the chloroplast. And then within the thylakoid, there's another fluid. We call that the thylakoid space. Not the fluid, but the space between it. Okay? And these are the structures within the chloroplast. So why do you think this thylakoid, or this grana, is folded over on itself? And what does this remind you of?
Surface area.
Yeah, so you increase surface area. Embedded within the thylakoid are the pigments and the enzymes and the electron acceptors and donors, which are going to complete the photosynthesis process, or at least the light-dependent reactions. And so if you have more membrane, you have more opportunities to photosynthesize more efficient. Okay? Good. And these are packed full of these grana, and all the necessary components are then within the stroma to complete photosynthesis. Okay, so in order to understand what's going on, we have to talk about three different components. You have a reaction center, an electron donor, and an electron acceptor. And these are going to pass electrons from one to the other. But they can't do that without first having energy. Okay, so this here is a photon. A photon is a packet of energy or packet of light which comes from the sun. And what will happen is when it gets to this electron, it can excite that electron to a higher energy state.
Oh, cool.
Yeah, nifty, right?
Yeah.
So now that this electron is at a higher energy state, it can be accepted by this electron acceptor. And now there's no electrons in there in the reaction center. And so this electron donor then, now because there's no electron in there, it can donate an electron into that lower energy state within the reaction center. So in the end, you'll end up right where we started. With an electron here. Where that donor put it, and then that acceptor can donate to somewhere else. Yes?
Where does the donor get its electrons?
Okay, so the donor, it depends on the chain of events. And it'll make more sense in the next one. All right, so if we want to number these, this happens first. The electron is excited in the reaction center to a higher energy level. This part happens second, where the electron is donated to the acceptor, and then the hole is filled by the electron donor. And so when something accepts an electron, it goes through a reaction, which we're familiar with. What do you call it when something receives an electron? It is being reduced, right? Because it's gaining a negative electron, so it's being reduced, right? So the donor is being oxidized, and the receptor is being reduced. And so we have then redox reactions occurring. Yeah, this is a redox reaction between the reaction center, a donor, and an acceptor. And then you'll have this same event happen multiple times throughout our light-dependent reactions in photosynthesis. Okay, any other questions?
I have a quick question.
Yes?
I'm just trying to see this in a bigger picture. What is this all doing inside of it?
Okay, we'll get to that next. Yeah. Yes. Okay, so the next thing is we're going to draw a Z diagram. So we're going to keep it simple in this one, and we're going to talk about different electron acceptors and donors. And we're going to start with, actually, I want to start with the green one. We're going to start with photosystem 2. Remember from the quiz, it's a little bit backwards, but that's what it is. Okay, and where do the electrons come from? Where they come from water, and you need a photon. Okay, what that photon allows photosystem 2 to do is to break off electrons and protons, and then what you're left with is oxygen. Okay, so this is why our photosynthesis reaction, on one of the products you have oxygen. Oxygen isn't used in any other steps in photosynthesis, but at the beginning we create oxygen when we split water into electrons, protons, and oxygen. Okay, so photosystem 2 then excites these electrons. So in this reaction, water is being oxidized, right? And photosystem 2 is being reduced. And then we're exciting these electrons, and now photosystem 2 is going to be oxidized. And our next chain of events is plastoquinone, is what it's called. And it accepts the electrons at the higher energy state. Okay, and there's actually if we write out an acronym for all of these, PPC, PPFN, okay? This is it'll make sense in the end. But we've gotten photosystem 2, and it passes it to plastoquinone, and the next one then is the cytochrome complex. And then the next one is plastocyanin. And at this point, our electron has been passed for so long, it's out of energy. It's a tired little guy. So we want to get him excited again. So luckily we have another photosystem further downstream which can take a photon and excite that electron to a higher energy level again. All right, so we're at PPC, PP. The next one is ferredoxin. And then the last one is Nadp reductase. And what that does is what? It's in the name. Reduces NADP plus to NADPH. So the fate of that electron is it is attached to NADP plus to make NADPH. Okay, and so this is what's called a Z diagram. Even though it's more like an M, it's called a Z diagram. All right, but if you can remember then, PPC, PPFN, I know these are large words. Some of them have reusing synonyms or prefixes, plastoquinone and plastocyanin. Another thing that the cytochrome complex does is when it passes that electron, it also uses that energy to pump protons through. And so now we have a concentration of protons. What did we do with the concentration of protons in the mitochondria? Okay, we had concentration gradient, and what did we do with that? Right, so this allows you to run these through ATP synthase to make ATP. And what was that process called? Chemiosmosis. Okay. All right, so this is the simple diagram. This is the simple one. Okay, we're gonna do it again, but add a little more detail on this next one. Okay, so these black lines represent our membrane. What was the membrane called? The what? Cytochrome membrane? Nope. Thylakoid, yes, good. So the thylakoid membrane, this is where all of these Z diagram participants are located. All right, so in order to get this started, though, of course, we need energy from the sun and little photons. And you have within the photosystem two sections. So one is called the light harvesting complex, or what is the other name for that? It's in the quiz. The antenna complex, good. And so these pigments are mostly chlorophyll B. And they can take that energy and pass it to each other. And sometimes that energy just goes off and is lost as heat. But if they can pass that energy to these chlorophyll A molecules, in the reaction center, chlorophyll A can split water into electrons, protons, and then oxygen, which we don't need. Yes?
So chlorophyll B is these little guys in here, and chlorophyll A is in here?
Yes, right? So if you wanted to label them, we'll label this at the end, but the ones on the outside are part of our antenna complex, and then the ones in the middle are our reaction center. And so now these electrons are excited. We're just going to do one at a time. So just like you guys before biology, you're at a higher energy level, right?
Yep.
All right, what was the next electron acceptor? What letter did it start with?
P.
Good. All right, so we got plastiquinone. It takes that excited electron. And then the next one started with, no, photosystem 2 was our first P, right? Good. C, the cytochrome complex. And it takes that electron, and it also takes protons and pumps them through into the thylakoid space. All right, what's the next one start with? P. Yep, you got plastocyanin. And now it's out of energy, right? Yeah, so cytochrome complex takes the energy from the electron and pumps hydrogen into the thylakoid space.
Okay.
All right, but now we can take another photon, pass it around. If we get it to these chlorophyll A molecules, we can excite the electron again, pass it to ferredoxin, and then use that electron to reduce NADP plus to NADPH in the enzyme Nadp reductase.
Reductase or reductase?
ACE. Should be ACE. ASE. Okay, so again, in the photosystem, you've got two parts where the light is harvested. You have the antenna complex, and then where chlorophyll A is there, you have the reaction center. But now what we've created is a concentration gradient of all these protons, right? And so just like we did in the mitochondria, we can use ATP synthase to convert ADP into ATP as they pass down their concentration gradient. Okay, and this process is called chemiosmosis, just like before. All right, so now we have some potential energy. Where is our potential energy in this? Yeah, so NADPH. And then where else? ATP, right? And so we can use these now in the light independent reactions, which we'll talk about on Monday, to fix carbon dioxide into bound carbon molecules together, and then you can bind those together to make sugars. Okay? All right, so what's great about this is it tells a story. All right, so you can go from beginning to end, similar to what it did in the mitochondria. And it's a great story, right? You'll probably see a Netflix series about this soon, right? Yeah, question? Light independent reaction. Okay, so these are the light dependent reactions. You have to have light in order to excite those electrons so then you can pass them down. But the next ones you can do without light. They're just, we're just taking carbon dioxide and fixing it.
So on the light independent one, it has to have light?
No, the light dependent one has to have light. So these ones have to have light. The next one does not. Yeah.
This whole thing is called the light dependent reaction?
Good, yes. This is all light dependent reactions. We drew the Z diagram, so sometimes referred to as that, which is part of it.